Hey everyone, Professor Spencer here. Welcome to this video on second messenger systems and signal transduction pathways. This video builds off the information in the video about the chemical classes of hormones. It pertains specifically to water-soluble hormones, so I highly recommend you watch that video first before you get into this one. In this video, we'll take a look at two very common second messenger systems, or signal transduction pathways, that are water-soluble hormones use to trigger functions within their target cells. These two pathways are the adenylate cyclase pathway and the phospholipase C pathway. We'll also discuss what the second messengers of each pathway are. At the end of the video, we'll do a short quiz to test your knowledge. Okay, let's get started. So the first thing to understand right away is that a hormone is what's known as the first messenger, and down the pathway we'll encounter our second messenger or messengers. Let's say, for instance, we skipped lunch today and we need to raise our blood sugar. To hold us over while we wait for food, the pancreas secretes the peptide hormone glucagon into the bloodstream. The glucagon circulates to the liver where it can bind to our liver cells, or our hepatocytes, and tell them to break down glycogen into glucose. So how could glucagon do this? Or how could any water-soluble hormone, for that matter, send a signal inside the cell? Well, it all starts with what's known as a G protein. You'll notice that adjacent to our membrane receptor on the intracellular side, we have a protein labeled G. This represents our G protein, and it's currently in its inactive state. When it's inactive, it just hangs out near our membrane receptor waiting to take action. This G protein also has a molecule bound to it, which is currently GDP, or guanosine diphosphate. Now let's bring our glucagon in. Our glucagon comes in and binds to the receptor, which causes the receptor to undergo a conformational change. Basically, this means that the structure of the receptor changes. So when it undergoes this change in shape, the GDP molecule on the G protein is bumped off. Now the G protein has a vacant space where that molecule was, and this space gets occupied by GTP, or guanosine triphosphate. GTP is what activates the G protein and causes it to move along the cell membrane. You can think of GDP as the G protein's lock. When you remove the lock, you can add the power source, which is the GTP. You can see that now we've got our movement happening, and we're one step closer to getting the hormone's message further into the cell. So where is this G protein going? It's going off to activate membrane enzymes, and two of the most common membrane enzymes happen to be adenylate cyclase and phospholipase C. And these enzymes help to facilitate all kinds of cellular functions. It's important to know these two common enzymes and the second messengers of each pathway. Let's talk about the adenylate cyclase enzyme first. When adenylate cyclase is activated by this G protein, it's stimulated to increase the amount of CAMP, or cyclic adenosine monophosphate, within the cell. This CAMP is our second messenger. CAMP is required to activate the next enzyme in the pathway, which are protein kinases. The more CAMP we have, the greater the chance we have of activating protein kinases. And it's these protein kinases that finally activate the functions that the hormone was trying to activate in the first place. So in the case of this hepatocyte, it's activating the enzymes that break down glycogen into glucose. The important point here is that the purpose of activating adenylate cyclase is to increase CAMP within the cell. And again, CAMP is our second messenger in the adenylate cyclase pathway. Let's talk about our second common pathway, which uses the phospholipase C enzyme. Just like we saw with adenylate cyclase, this enzyme also has to be activated by our G protein. So GDP is bumped off and then replaced with GTP, and the G protein runs along the cell membrane and activates phospholipase C. Once activated, phospholipase C splits phosphatidylinositol bisphosphate, also known as PIP2, into diacylglycerol and inositol triphosphate, or IP3. These two products of this reaction are the second messengers of this pathway. Diacylglycerol can then participate in functions like lipid metabolism, while IP3 can help adjust calcium levels within the cell. Just so you're not overwhelmed, you will likely not need to recall the names of these molecules, just their abbreviations. Again, diacylglycerol and IP3 are the second messengers in the phospholipase C pathway. Now you might be wondering, okay, all this is cool, but how does it stop? This process will keep occurring as long as the hormone is bound to the receptor or that GTP is bound to the G protein. Eventually, our glucagon will degrade and GTP will be hydrolyzed back into GDP, and this resets the whole system. That way, our liver cell here doesn't just continue to break down glycogen into glucose forever. Before we go any further, here's a simple chart for your notes. The chart shows that indeed, we need G proteins to be activated before anything else can occur. The G protein moves along the membrane where it can activate our membrane enzymes. These enzymes can be adenylate cyclase or phospholipase C as two common examples. Adenylate cyclase raises the amount of CAMP within the cell, and CAMP is our second messenger. Phospholipase C splits PIP2 into diacylglycerol and IP3. These products are the second messengers of the phospholipase C pathway. Put another way, the second messenger of each pathway is what's created when the enzyme of that pathway is activated. Okay, I know that was a lot of information. Before we take the quiz, feel free to watch the video as many times as you need. If you're ready, let's take the quiz. These questions are based on what you'll probably see on a multiple choice or fill-in-the-blank exam. What is the name of the protein that activates membrane enzymes? G protein. What needs to happen first to initiate a signal? Water-soluble hormone binds to the membrane receptor. CAMP is the second messenger of this pathway. Adenylate cyclase. Diacylglycerol and IP3 are second messengers of this pathway. Phospholipase C. Again, if any of this was confusing, don't feel bad about needing to watch it again. This is one of the harder concepts to wrap your head around with the endocrine system, so repetition is key to grasping it. That's all for this lesson. I'm Professor Spencer, and I'll see you next class.